Welcome. In this session, I'm going to start a neat objective on the chapter biological classification. The first question from level 1, all eukaryotic unicellular organisms belong to. So, here the first group of organisms, Monera. Monera have given rise to Protista, and this group Protista it diversified to form fungal group and plants and then animalia. So here the Monera it includes all unicellular prokaryotic organisms. So it includes all unicellular prokaryotic organisms. While coming to Protista, this includes all unicellular eukaryotic organisms. So all eukaryotic and unicellular organisms were included under the kingdom Protista. Right. Coming to fungi, it will include unicellular. Okay, if you are unicellular and majority of them are multicellular. So we find unicellular or multicellular eukaryotic organisms. So eukaryotic organisms. And coming to plantae, this group includes also unicellular or multicellular eukaryotic organisms. Okay, eukaryotic organisms. And lastly, the animalia, it will include only multicellular eukaryotic organisms. Okay, so here the question is all eukaryotic unicellular. So all eukaryotic unicellular organisms belong to, okay, that is in case of Protista, all unicellular eukaryotic organisms, okay, so they belong to the group Protista. So the right answer is option B, Protista. Next question, contagium vivum fluidum was proposed by. So here contagium vivum fluidum. So in this case, it means the contagium stands for infectious, and vivum it stands for living, and fluidum this stands for fluid. So the meaning of contagium vivum fluidum is infectious living fluid, and that was actually given by M.W. Bejernik. Right, so this statement is given by M.W. Bejernik, whereas D.J. Ivanovsky given the term virus. Okay, and then W.M. Stanley. So W.M. Stanley, he showed that viruses can be crystallized, can be crystallized. Okay, right, and Robert Hooke. So this person discovered cell in 1665. Of course, he discovered cell in a thin slice of bottle cork, in thin slice of bottle cork. Okay, so he discovered cell for the first time in 1665. So the contagium vivum fluidum was actually given by M.W. Bejernik. So right option B is the right option. Next question, difference between virus and viroid. Okay, so we know that this virus is composed of proteins. Okay, so that is protein coat, and also it consists of either DNA or RNA. Whereas in case of viroids, so the viroids are composed of made up of only single-stranded RNA molecule. So there are no proteins present in case of viroids. So the difference between viruses and viroids is the absence of protein coat in viroid, okay, but present in virus. So absence of protein coat in viroid, yes, and there is this protein coat present in case of virus, okay, right. Presence of low molecular weight RNA in virus. Okay, so here the single stranded RNA in case of viroids is of low molecular weight, is of low molecular weight. So low molecular weight RNA is present in case of viroids, not viruses. So this should be, okay, viroids, right. Both A and B will not be the answer, none of them. Okay, so the right answer, the difference between viruses and viroids, so that is option A is the right answer. Next question, organisms living in salty areas are called. Okay, so here the methanogens are bacteria that live in marshy areas and also present in gut of ruminant animals, present in the gut of ruminant animals. Okay, and then coming to halophiles, so these are the bacteria that live in salty areas, are the bacteria that live in salty areas. Then coming to heliophytes. So heliophytes are plants that are actually sun living, sun loving plants. So heliophytes are sun loving plants. So in this case, okay, so here the halophiles are bacteria, the methanogens are also bacteria, whereas heliophytes are sun loving plants, like example mango plant. Then coming to dermoacidophiles. So these dermoacidophiles are the bacteria that live in hot springs, that live in hot springs. Okay, so that is with high temperatures plus, okay, high acidic conditions. So those bacteria that live in these conditions, they are called dermoacidophiles. So the question, organisms living in salty areas. So here the organisms that live in salty areas are called halophiles. So the option B will be the right answer. Next question, naked cytoplasm, multinucleated and saprophytic are characteristics of. So in case of Monera, okay, so cell wall is present. So when cell wall is absent, okay, we use the term naked cytoplasm. Okay, cell wall is absent. Okay, so in case of Monera, cell wall is present except mycoplasmas. So except mycoplasmas. Okay, then coming to Protista, not all Protistans will have cell wall. Some of them will have cell wall and some will not have cell wall. Then in case of fungi, also cell wall is present. And in case of slime molds, okay, so there is no cell wall. Okay, cell wall is not present, and it will show a condition multinucleated or cenocytic condition, and these are saprophytic. Okay, so the right answer, that is option D. Next question, members of Phycomycetes are found in. So we know these Phycomycetes are grouped under kingdom fungi. So these are, okay, so group of fungi or group under kingdom fungi. Okay, and let's see the characters of these Phycomycetes. Yeah, they live in aquatic habitats. Yes, this is correct. And they are also seen on decaying wood, and they are also seen in moist and damp places. And they also uh, seen as obligate parasites on plants. So this is also correct. So here choose from the following options. So all of the mentioned characters are true. So hence option D is the right answer. Next one, which of the following categories of organisms do not evolve oxygen during photosynthesis? So here the organisms that, okay, do not evolve oxygen during photosynthesis are said to exhibit, okay, anoxygenic photosynthesis. Okay, so they perform anoxygenic photosynthesis. Perform an oxygenic photosynthesis. Okay, so let's see the options. Red algae. So red algae, they are plants and they perform they perform oxygenic photosynthesis. They perform oxygenic photosynthesis. And coming to photosynthetic bacteria, so they perform an oxygenic photosynthesis. And then the C4 plants with the Kranz anatomy, they also perform oxygenic photosynthesis. So here oxygenic photosynthesis, which means there will be release of oxygen in the process of photosynthesis. So here an oxygenic photosynthesis, which means they do not release oxygen during photosynthesis. So they don't release oxygen during photosynthesis. So oxygenic photosynthesis, which means they release oxygen during photosynthesis. Okay, so in C4 plants also they perform oxygenic photosynthesis. Okay, so C4 plants are the one who show C4 pathway, so that we will discuss, okay, it later. So already we know, we studied in photosynthesis in higher plants, okay, about the C4 plants. Then coming to blue green algae, so in short we call BGA, also called cyanobacteria. So these cyanobacteria, they also perform oxygenic photosynthesis. So the organisms that do not evolve oxygen during photosynthesis, they are photosynthetic bacteria. So that is option B is the right answer. Next question, in prokaryotic cells. So in prokaryotic cells, okay, internal compartments are absent. Yes, okay. So in prokaryotic cells, internal compartments are absent. Okay. So here internal compartments like we see in case of eukaryotic cells. Okay. So there will be, okay, two compartments due to presence of nuclear membrane. So this will be cell membrane and this will be the nuclear membrane. So due to presence of this, this will act. So here outside the nuclear membrane is one compartment and inside is another compartment. But coming to the prokaryotes, we don't see, okay, the nuclear membrane, hence no internal compartments will be seen in case of a prokaryotic cell. So that internal compartments absent is right. Then nucleus is absent. We know prokaryotic cells don't have nucleus. This is also right. Then ribosomes are of 70s type. Yes, so ribosomes are of 70s type. So in case of eukaryotes, so in eukaryotic cell, the ribosomes will be of 80s type. So the ribosomes are of 80s type, and in prokaryotes it will be 70s type. So in prokaryotic cells, so all these options are right. So option D is the right answer. The next question, match the type of bacteria listed. Okay, so the type of bacteria listed in column 1 with their activity, okay, given in the column 2, and okay, choose the correct combination, right? So here the first one, streptomyces. So here these streptomyces are bacteria, okay, and they are actually the source of antibiotics. From them we obtain antibiotics. So here, okay, Q. Then rhizobium, so these are also symbiotic bacteria that actually help in uh, fixation of nitrogen. So hence, so the R, that is nitrogen fixation. Then nitrosomonas, so this nitrosomonas is a bacterium that actually helps in nitrification, so this nitrification is a conversion of ammonia into nitrite. So we'll see here, okay, so 2NH3, okay, so NH3, I'm sorry, 2NH3 plus, okay, 3O2 will result in formation of 2NO2 minus. So this is the nitrite, okay, plus 2H plus and 2H2O. So this is done by, so that is formation of nitrate is done by the nitrosomonas, done by nitrosomonas. Okay, of course, one more bacteria can also help in this process, is called nitrococcus. So nitrosomonas or nitrococcus. So these two can help in conversion of ammonia into nitrite. So this process, of course, nitrite will be later converted into nitrate. So those two steps together we can consider it as nitrification. So nitrosomonas will help in nitrification. Then the next one, acetobacter. So acetobacter aceti is used in manufacturing of acetic acid. Okay, so this vinegar is actually aqueous solution of acetic acid, is aqueous solution of acetic acid. Okay, so here out of the options, so D option, so AQ, BR, CS and DT, so that is option D is the right answer. Next question, which of the following are true archaeobacteria? So here the archaeobacteria are actually extreme halophiles. The true archaeobacteria are extreme halophiles. They are extreme thermophiles or thermoestophiles, and even the methanogens. So halophiles, thermoacidophiles, and methanogens are actually true archaeobacteria, and they uh, don't have peptidoglycan in the cell wall. So here presence of peptidoglycan cell wall is given. So hence will not consider this statement. So the 1, 2, 3 are the right characters of true archaeobacteria. So option A is the right answer. Next, select the correct combination. So select the correct combination of the statements regarding the characteristics of certain organisms. Okay, so methanogens are archaeobacteria, yes, which produce methane in marshy areas. Yes, this is right. Then nostoc is a filamentous blue-green alga which has capability to fix atmospheric nitrogen. So with the presence of heterosis, yes, this is also the right statement. Then chemosynthetic autotrophic bacteria synthesize cellulose from glucose. No, so they don't have cellulose in their cell walls. Okay, and they don't synthesize cellulose from the glucose, so that is a wrong statement. Then mycoplasma lack cell wall, and they can survive without oxygen. Yes, this is also a right statement. So A, C, okay, sorry, A, B, D are the right statement. So hence the option D, A, B, D is the right answer. Next question, which one of the following statements is correct? So here all bacteria are heterotrophic. No. Okay, so there are heterotrophic, there are autotrophic. Okay, under the autotrophic also we find photoautotrophic and chemoautotrophic. So here, okay, so we find when coming to nutrition, okay, in bacteria, so they have autotrophic and heterotrophic. Under autotrophic we see both photoautotropic bacteria and also chemoautotropic bacteria, right? So bacteria are either heterotrophic or chemoautotropic. So this is also a wrong statement. And then bacteria are either photoautotropic or chemoautotropic. Okay, so this is also a wrong statement. So bacteria can also be photoautotropic. Yes, this is a right statement. So out of these four, option D is the right answer. Next question, the thalloid body of a slime mold. Okay, so the group of slime molds, or the class of the slime molds called, okay, myxomycetes, right, is known as, we know here, mycelium. So these slime molds, we don't consider them as fungi. So these slime molds will be coming under kingdom Protista. So they are under kingdom Protista. And mycelium is the body of fungus. So it is the body of a fungus. Okay. Then coming to protonema. So the protonema is the first stage, okay, in the life cycle of moss. So protonema is the first stage, okay, in the life cycle of moss plants, bryophytes. So they are bryophytes. So that is this protonema will develop from the spore. So that is spore germinates to give rise to this protonema stage. Okay, so the next one, coming to plasmodium. Yes, this plasmodium, okay, right, is the thalloid body of slime molds. So that will be, okay, having a amoeba like shape, and then it has, okay, many nuclei. Okay, so this is the thalloid body of slime mold. And fruiting bodies are structures that actually produce spores, so which can also be seen developing in case of these slime molds. Okay, so the thalloid body of a slime mold is actually the plasmodium. So that is option C is the right answer. The next question, match the following and choose the correct combination. Okay, Chrysophytes. So here Chrysophytes, the examples for Chrysophytes include diatoms. So, so diatoms. Then dinoflagellates. The dinoflagellates example is Gonilox. Then euglenoids. So the example for euglenoids group is Euglena. So all these are different groups under kingdom Protista: the Chrysophytes, dinoflagellates, euglenoids, and protozoans. So all these are, okay, groups under kingdom Protista. So protozoans, okay, one example of protozoans is Paramecium. Okay, so 4 3 2 1, so that is option A is the right answer. The next question, which of the following is a flagellated protozoan? So here a flagellated protozoan is a one. Okay, so a protozoan having flagella, right? And these flagella are locomotory structures. So they are locomotory structures. So in case of amoeba don't have flagella. Instead, Okay, so they have pseudopodia for locomotion. So pseudopodia for locomotion. And even in case of entamoeba also have pseudopodia. Okay, next coming to trypanosoma, you can see here a picture trypanosoma gambiense. So this causes disease. So trypanosoma causes a disease called African sleeping sickness. Causes African sleeping sickness. And you can see in this picture, so there is a flagellum. Okay, so all along the body you will find the flagellum, right? So it is in the trypanosoma we find the presence of flagellum. And coming to plasmodium, in plasmodium, okay, no locomotory organelles are present or locomotory structures. So hence the flagellated protozoan out of all these four is a trypanosoma. So that is option C is the right answer. The next question, among rust, smut and mushroom, all the three are. So here the rust and smuts, these two are parasites. Okay, so while mushroom is saprophytic, okay. So all the three are pathogens. Okay, no, so mushroom is not a pathogen. Okay, so this is wrong. Then all the three are saprops. No, so rust and smuts are parasites and mushroom is saprophytic, so this is also wrong. They bear ascospores. No, so ascospores are seen in case of the group Ascomycetes. And here the rust, smut and mushrooms, all these three, they belong to the group Basidiomycetes. So they belong to the group Basidiomycetes, and Basidiomycetes will have basidiospores, that is they bear basidiospores. So the right option is option D. So with this I will end today's session. So in the next session we will see a few more questions from level 1, and then we'll see the questions from level 2